want to understand the way to achieve healthy longevity by triggering adaptive stress responses. And we use many kinds of different stresses, such as heat, such as ultraviolet light, microbiome, circadian clock disruption. And we also research a very big range of adaptive stress responses, such as mitophagy, metabolic plasticity, proteasome. The aim is to find interventions that can extend the healthy period of life by triggering adaptive stress response.
So I'm investigating the role of Pseudomonas as a species of bacteria in longevity. We saw that there was a particular gut microbial signature that existed in the microbiome of centenarians. We don't know if the bacteria that exist in this signature are the cause of the phenotype or the consequence of long life. So the one that we chose is the bacterium from the genus Pseudomonas. In my project, I investigate the interaction that this bacterium has with the host. And as host, I'm using these tiny worms called Cynorhabdotus elegans. In the lab, what I'm doing is, after finding some particular strains of Pseudomonas that extended lifespan of the host worms, I am now extending the research to see if they are also capable of extending the health span. And for this, we have different assays. We look at the strength of the host after being exposed to this bacterium. We also look at their neuromuscular capacity, because we know that these parameters are declining during aging, right? We are, of course, not as strong as we were when we were 50 years old, if you look at the 80-year-olds.
Our lab basically talks about stress adaptation, and this is when you give moderate stress to an organism and it boosts its functions in a way that it brings the organism into a balance. So this is the same idea behind my experiments. We are using something that in moderate dose, such as ultraviolet light, to boost the functionality of the organism and see afterwards how the stress adaptation happens. One of the profound methods that we're using in the institute and also in our lab is using the core facility of proteomics, where we basically treat our worms with untreated group and treated group with the ultraviolet to understand how the molecular pathways and how the proteins are behaving when we are giving them the stimulus of ultraviolet. And this is a very profound step because it gives us a very interesting insight to understand in the molecular level what are those changes and go about it with experiments. We are basically found out by our experiments that it influences the mitochondria, which are the powerhouses of the cell, which are very important for our energy production. What happens is that in young organisms, mitochondria manages to boost its functionality by using different pathways, different processes in the body, and eventually bring the body to the energy homeostasis, meaning energy equilibrium. And that's how the body becomes so stress resistant and so stress adapted to the ultraviolet light.
Interestingly, the same kind of lamps, these UVB lamps that we tried in this project, they are already used in humans to cure skin disorders. So they are already proven to be safe in humans to use in the clinics. So we now have an existing patent application with one of my students, Asia, where we want to actually now test these lamps as dietary restriction mimetics to promote healthy longevity, because these DR mimetics are well known as very effective treatments for healthy aging. So basically, we are very fast, I think, in going from the bench to the clinics as much as we can.